polypeptide longer than 100 amino acids, and in some cases, a protein can consist of more than one polypeptide chain. Okay, more than one. We'll talk about a couple of examples of that. So this is a table from the, I think, I don't know if it's from this book. I think it's from a previous version of the Campbell book. This version of Campbell has a nice sort of 2-page spread on this, but this summarizes some of the different kinds of functions that we observe for proteins. And it's just important for you to know some of the different kinds of things that proteins can do. As we said, they can function as enzymes, and that means that they can accelerate the rates of chemical reactions in cells. Absolutely critical for life, right? We have to have that kind of function. But we also have to have a lot of these other functions, such as support for the cell, and these include things like collagen, elastin, structures that are found in our skin and in feathers and other kinds of structural features that we see in animals and plants. Proteins are also important for storing amino acids, excess amino acids. They're important for transporting substances such as oxygen and other kinds of smaller molecules. They sometimes function as hormones. They can function as receptors for signaling in cells. They can help cells to move around, and they also help defend cells against pathogens. They help defend against disease. So all sorts of functions for proteins. So as you can imagine, with that kind of incredible variety of function, there's been a huge effort over the past several decades, and really probably even before that, to really understand what are proteins, what are they made of, what do they look like, and how do they work, right? And so let's just talk a little bit about the chemistry of proteins. So this is a diagram of a, what is that? Amino acid, thank you. Yes, it's amino acid. And it's called amino acid because it has a central carbon molecule here that's hooked up to an amine group on one side and carboxylic acid on the other side. Okay, this carbon is called the alpha carbon. And very importantly, it also has an R group connected to it. And depending on what the chemistry of that R group is, that makes this an amino acid that's either alanine or phenylalanine or tyrosine or et cetera, one of the 20 common amino acids. Okay, so those are defined by what this R group is. And these amino acids will differ in their chemical properties based on the property of the side chain. Now, what I'm showing you here are the 20 amino acids classified according to their chemical properties. All right, so there's a set of amino acids shown at the top that are nonpolar. Okay, so nonpolar, so that means that they tend to what? They tend to hydrogen bond? No, right? They tend to not hydrogen bond, right? So these tend to make hydrophobic kinds of interactions, right? And then we have in the middle, we have a set of amino acids that are polar, right? And so these, if you look carefully at these chemical structures, you can see that, so we have some OH groups here, we have an SH group here. So these are going to be groups that have polar properties, such that they can form hydrogen bonding interactions with each other or with other amino acids or with water, of course, or other kinds of molecules, okay? So that's polar amino acids, and then we have the charged amino acids down here, and these can either be basic or they can be acidic. So, as I said before, I don't expect you to memorize the chemical structure of each amino acid such that you could, say, draw it on an exam. You can if you want to, but I do want you to know that there are 20 amino acids. I want you to know the properties of these amino acids such that if I showed you a sequence of a polypeptide and I said, is this polypeptide likely to form hydrogen bonding interactions, you can answer that question, that sort of thing, right? So just the basic chemistry of these, and I think if you know your basic general organic chemistry, you can answer that question. All right, and so how do these come together? So again, this is a nice example of a dehydration reaction where these growing polypeptide chain, which is shown here, is being linked to an incoming amino acid with the loss of a water molecule to make a polypeptide that's one amino acid longer. Okay, and so and very importantly, this bond right here between this carbonyl carbon and the adjacent nitrogen of the next amino acid is called the peptide bond. Okay, so it's called the peptide bond right here. And it has some very important properties. We might talk about those a little later, but I do want you to know what that bond is. So if I showed you a drawing and I said which bond is the peptide bond, you would know that that's the bond that we're talking about. Okay. And another very important thing that I want you to know is that we can sort of draw and think about polypeptides according to the different features of the chain. For example, these atoms right here, they're shown in the boxes, are called the backbone. Right? These are the backbone atoms. Does anybody know? So why do we call those the backbone? Anybody? So basically, if you look at the drawing, you'll see that the backbone is basically the same, right? As we build up this polymer, the backbone is the same for every amino acid. What's changing is the side chain over here. Okay, so these are the side chains. So these backbone atoms are common to all polypeptides, but each polypeptide, depending on what the amino acid composition is chemically distinct based on the properties of the side chains that are in that particular polypeptide. Okay, so if you think about it, there's a vast number. You guys could just try to do the back of the envelope math. How many different kinds of polypeptides of 100 amino acids in length could you have? And the number is big, right? It's really big. So biology for sure does not explore every single possible flavor of protein that could possibly be made, right? There just isn't enough matter, I think, in the universe to do that. Okay, so biology has really, as organisms have evolved, there are very specific types of proteins that we see and types of protein structures that we see used over and over again. And the reason is they work, right? They work. And so biology is a very utilitarian kind of thing. So basically, if a molecule is functional and it works well enough, then it tends to be used over and over again. So once these polypeptides are linked together, they form these long chains. And what's really important to know is that these long chains then fold up into three-dimensional structures. And it's the three-dimensional structures of proteins that give them the properties that they have. So if you have a polypeptide chain and it's in a denatured state, that means a unfolded state, and we might be able to induce that in the laboratory, for example, by putting proteins into a solution of an organic solvent or a very greasy, like a detergent type solution that would disrupt the kinds of interactions that are required to form a three-dimensional structure of a protein. Typically, under those sorts of artificial conditions, those proteins won't be functional. Okay, and the reason they're not functional is that they're no longer forming the kinds of three-dimensional structures that we see here. They're so essential for proteins to work. This is showing, these are a couple of different kinds of representations of proteins. By the way, how do scientists know the structure of proteins? Anybody know? X-ray crystallography. What's another way that we know the structure of proteins? I think I heard NMR. Nuclear magnetic resonance is another way that we know the structure of some proteins. Yeah. And actually, in some cases, we actually understand the structure of proteins from other more indirect kinds of experimental methods. But in general, when I show you, or when you see a structure of a protein that looks like this, and it's an actual representation of a protein structure, that structure in general has been determined by X-ray crystallography. Question? Great question. So he's asking, is it possible to infer the three-dimensional structure of a protein just based on looking at the sequence? That's a fascinating area of very active research. So people would love to be able to predict accurately the protein, fold of a protein based on just looking at the polypeptide sequence. In some cases, it's been possible to do that. It turns out that it's very challenging, though, because there are just so many potential ways that a polypeptide chain could fold up. Okay, and let's to sort of understand that a little better, let's look at a little bit more about how these structures actually come together. So there really are 4 levels of protein structure that we can consider. Okay, so these are described in a lot more depth in chapter 5 in your book. But very briefly, these include the primary structure of the protein. So the primary structure just means the order in which the amino acids are linked together. By the way, I think I didn't say this on the last slide, but please notice and please be aware that because of the way that these amino acids come together, a polypeptide chain has a polarity to it, right? It has one end that's what? What's on one end? The amino group, right? So it's shown right there. So we have the amino terminus, the N-terminal end of a protein. What's on the other end? Yeah, the carboxyl group. So we have one end that has an amino terminus, and we have another end that has a carboxyl terminus. Okay, so we have N-terminus and a C-terminus. And so that's the primary structure. And then this chain then folds up into what's called the secondary structure. There's 2 primary types of secondary structure that we see in proteins. One is called an alpha helix. The other is called a beta pleated sheet. And if we look at how those types of secondary structural units are put together in proteins, they involve interactions between the backbone atoms of the polypeptide chain, okay, the backbone atoms that we just talked about. And they tend to be, these are not covalent interactions most of the time. Okay? These are usually weak interactions like hydrogen bonding interactions, but there's so many of them, there's so many hydrogen bonds that those can actually be very, very stable units of secondary structure. Okay? So alpha helices and beta pleated sheets. And then those units of secondary structure fold up further to form what's called tertiary structure. And in tertiary structure of proteins, those tertiary structures are stabilized generally by the interactions of the side chains. Okay? So for example, when we try to predict protein structure, we actually have to look at the side chain interactions that could occur and then try to predict which ones actually do occur. Okay? And there's lots of research being done to try to do that more accurately than is currently possible to do. And then finally, we have a structure over here called quaternary structure. So in some cases, a single polypeptide that folds up to form a tertiary structure like this will then interact with additional polypeptides that are folded up to form their own tertiary structures to form a much larger structure called a quaternary structure. And again, those interactions will typically involve not covalent bonding interactions, but non-covalent interactions like hydrogen bonds, as we said, but also van der Waals interactions and sometimes hydrophobic interactions as well. Okay? So what do these structures do and what do they look like? Well, so here's, again, a couple of examples of quaternary structures. So collagen, which is one of the central proteins that's very important for the structure of our skin, our connective tissue, and things like that. This is actually a molecule that's formed from 3 individual polypeptide chains that come together and form this fibrous kind of structure. And then over here is another example of a quaternary protein structure, hemoglobin, which is important in our blood for carrying oxygen. These are showing, in this cartoon, individual molecules of heme that are bound in each of the 4 polypeptides that come together to form an active hemoglobin molecule. So hemoglobin is not active unless it has all of these components together and they're folded up properly, and they have the heme molecule bound, which can then interact, has an iron in the center, which then binds to oxygen. And as I said before, so you might wonder, well, how do we know all these gorgeous, how do we know these gorgeous structures? How do we know what they look like? And it turns out that in many cases it's been possible to crystallize proteins. And so as soon as scientists back in the 60s and 70s started figuring out that they could actually isolate and purify certain types of proteins and make them very, very pure, remove other contaminating molecules, then it became possible in some cases to induce these samples of purified protein to crystallize. And in a crystal, it's just like an organic chemistry lab, right, where you make a crystal of a small molecule, an organic molecule. With macromolecules, it's the same idea. You basically have a molecule of protein that's packed against another molecule of the same protein, and they typically have the identical structure, and that forms a very large lattice, 3-dimensional lattice. And so when that lattice is exposed to X-rays, and does anybody know, I'm just curious, does anybody know why do we use X-rays to do this? Why not other wavelengths of radiation? Anybody know? I think I might have heard it. So X-rays basically have a wavelength that's very similar to the length of the bonds that are found in these macromolecules. Okay, so it's very appropriate for looking at the structures because that's the sort of the length of bond that we're trying to detect, right? So we use X-rays. Here's the crystal, and so typically this is done by having some kind of X-ray source that is applied to the crystal. There's a diffraction pattern that one can then measure, and eventually, through various computational methods, calculate a structure of the molecule that's consistent with the diffraction pattern that we observe. So it's a very indirect kind of experiment. Right? We're not directly observing the structure of the protein like you might do in microscopy, but we're actually getting a very indirect measure of the structure of the protein based on its diffraction pattern in this kind of an experiment. And in many cases, those types of experiments can lead to the ability to actually look at a model of the protein structure and understand in molecular detail how it folds up and how it binds to other molecules. Like in this example right here, where you see a molecule of an enzyme called RNA polymerase, whose job is to synthesize a piece of RNA that is that has a DNA template, which is right here. We understand a lot more about how that kind of process works when we have these sorts of detailed structures available to look at. Okay, so with that, I just want to then briefly talk about nucleic acids. So you know that there are 2 kinds of nucleic acids, RNA and DNA, and the amino acid of a polypeptide is programmed by a unit of inheritance that's found in RNA or DNA, right? And we often call the units that encode proteins genes, and these are made up of DNA. And I just very briefly want to remind you that in a eukaryotic cell, we have cytoplasm, we have the nuclear membrane, which is right here, and in a eukaryotic cell, the DNA is in the nucleus, and a molecule of an enzyme that we just looked at, the RNA polymerase, will come along and copy one of the DNA strands to make a molecule of messenger RNA, which is shown right here. That gets shipped out of the nucleus, and in the cytoplasm, it actually gets bound up by ribosomes. These are the very large machines in the cell that are responsible for reading the code in the messenger RNA and synthesizing a polypeptide from that. So this is very quickly how polypeptides are made. We'll talk about this in more detail, of course, later. What happens in bacteria? Anybody know? In bacteria? Is there a nucleus? No. So in bacteria, this is all just going on inside of the cell, right? So there may be localized regions where protein synthesis is occurring, but there's not a separation like we see in eukaryotic cells like this. Okay, and so you know that nucleic acids are made up of nucleotides, and these have 3 components. a base, a sugar, and a phosphate. Okay, and so when we say the word nucleotide, we're referring to an individual unit that doesn't have the phosphate. That has the nucleotide means it has the phosphate. If we say nucleoside, it doesn't have the phosphate. Okay, and so the next slide just shows a diagram of this. So here's the way that these bases are put together. You have the base over here, the sugar. This is a pentose sugar, which is called ribose, and then we have a phosphate group over here. And these guys are linked together by the same kind of chemistry, right? Dehydration reactions that link them together. So here are the 5 bases that are found. So C, T, A, and G are found in DNA. In RNA, there's no T. It's actually a U, right? It's a uracil. So you should know that these are called pyrimidines, know that these are called purines. Again, you don't have to be able to draw the chemical structure, but you should know a little bit about the properties of these individual nucleotides. And we'll talk about, as we go through the, next week's lectures and beyond, you'll hear more about this, okay? So let's do a couple of quick iClicker questions. And the first one that I want to ask you is this question right here. Which group of large biological molecules is not synthesized by dehydration reactions? All right, so think about that. I'm going to start the timer. Everybody got their clicker out? Okay. I'm going to start the timer, and you'll have about 30 seconds to answer this, okay? So go ahead and go for it. Okay, so about 8 more seconds. So place your vote. 4, 3, 2, 1, stop. Okay, so let's see how we did and see if this is showing up. Oh, it's not showing up. Oh, shucks. Okay, so 74% of you voted for the answer to be B, which is fatty acids, and that's right. Okay, so as we talked about, that's the correct answer. Okay, and the next most popular answer was D. 18% of you voted for nucleic acids, but actually nucleic acids are also polymers, and they're also hooked together by dehydration reactions. Okay, and let's close that up. Okay, so just a couple more things. We're going to do one more iClicker in about 1 minute. Okay, I want to tell you one more thing before we do that. And this is about the DNA double helix. So DNA has 2 polynucleotide strands that basically form a double helical structure. And the reason that they form a structure like that is that the individual bases can pair with each other, right? So A pairs with T and G pairs with C. In RNA, A pairs with U, right? It can pair with U. And very importantly, these molecules are running in opposite directions to each other. So please take the time to look in your book about the synthesis of a polynucleotide strand, because you'll see that just like in a polypeptide, there's a polarity to this polymer, right? So it has, in the case of nucleotides, we have an end, we have a what's called a 5' end, and we have a 3' end. Okay, and that's depending on which part of the sugar molecule on either end, which carbon is actually exposed on the ends. Okay, so there's a polarity to these molecules, and when they come together to form a double helix, they're actually running in opposite directions. So the DNA molecule is called anti-parallel. It's an anti-parallel structure. Okay, so the 5' and 3' ends are running in opposite directions in the double helix, and this is a diagram that just kind of shows what I mean by that, right? So we have the 5' end here, and on the other strand we have the 3' end. So these are running anti-parallel, and you can see the bases pairing up with each other in the center. And this has a very important consequence for the way the DNA duplex can be copied by enzymes, right? And we'll talk a lot more about that later in my section. And so I want to just now, we have about not very much time. I want to go to the last iClicker question that I want you to answer. I'm going to give you a little bit longer to do this. So have a look at this question. Which of the following pairs of sequences could form a normal DNA double helix? And you know that these guys have to run in anti-parallel directions, and you know that A pairs with T and G pairs with C. So take 1 minute to do this one, okay? Okay, so 20 seconds. 10 seconds to go. Okay, 3, 2, 1, stop. Okay, let's see how we did. So great, 90% voted for D, and that's exactly right. Woohoo! See you guys on Monday. Yeah, I'll put the slides up.